day students. In our series of PCG412 lab chromatographic techniques, today we'll be studying isolation of purine alkaloids. Exercise 1, isolation of caffeine from tea, coffee and kola nuts. These are the what we'll be going through. This is the outline of what we'll be doing today. Objectives, an introduction. We'll go through the procedure. There's an exercise for submission on LMS which will be graded, conclusion and your reference. These are the objectives of the lab. You should be able to isolate purine alkaloid from plant materials containing them. You should know the purpose of adding different reagents in the procedure. You should be able to identify purine alkaloid using simple chemical text and to know the pharmaceutical uses of purine alkaloids. At the end of the lab session, students should be able to isolate purine alkaloids from plants containing them using appropriate chemicals and procedure, and identify presence of purine alkaloids in samples and know their medicinal uses. For a brief introduction, natural products can be isolated from plants and other natural sources using different techniques such as extraction, filtration, precipitation, distillation, and so on. Purine alkaloids are referred to as pseudo alkaloids because they do not behave chemically like other alkaloids and are not biosynthetically obtained as other alkaloids. Examples are caffeine, theophylline, and theobromine found in tea, coffee, and kola nuts. You have to find out the chemical structures and the botanical sources. In this experiment, we are going to be dealing with tea, and we are using top tea bags. This is a list of the equipment and materials and chemicals that were used in the experiment. For the procedure, first and foremost, 5 tea bags were opened and the contents weighed. 400 ml of water was boiled and the weighed tea bags were added to it, and it was brought to boiling by heating for 15 minutes on a hot plate with continuous stirring. Contents of the beaker were now filtered hot. The hot filtrate was mixed with basic lead acetate until no precipitate was formed again. The mixture was then heated to boiling and filtered rapidly using a Buckner funnel setup, as you can see on the slide, and I'll post the video on LMS for you to see and appreciate what I'm talking about. The residue remaining on the filter paper was also washed with boiling water and the filtrate was heated to boiling. Sulfuric acid 25% was added until precipitation stopped. This solution was filtered hot and allowed to cool. Then decolorizing charcoal was added to the filtrate. The filtrate with decolorizing charcoal was placed on low heat for about 15 minutes to ensure clarification. Now this was then filtered to get a clear filtrate, and it was allowed to cool. The clear filtrate was then partitioned in 3 successive chloroform portions. Now these 3 chloroform portions were combined and anhydrous sodium sulfate was added to remove any traces of water. The chloroform partitions were then placed in evaporating dishes and carefully evaporated to dryness at low heat. A small portion of the chloroform extract was transferred into a separate evaporating dish, and Mirror test was performed to confirm presence of purine alkaloid. You can see the steps there and you can see them in your manual. Upon evaporation, hot ethanol was added to the residue to effect a solution, and this hot ethanol was at 60 degrees centigrade. This was then transferred into a previously weighed beaker, and the solution of the ethanol and the crystals were evaporated to dryness or left overnight to recrystallize. The weight of the crystals obtained and percentage yield was calculated. So from these crystals we will use for the next two exercises, exercise 2 and 3. The video of the isolated crystals will also be placed on LMS for students to appreciate the crystals, their shape and their color. Now for the exercises for this experiment, you have to write a one-page report, preferably a PDF document, including a few sentences of what purine alkaloids are and their botanical sources. You have to tabulate the procedure of Murray test, observation, inference in one row. Brief discussion on the yield obtained in this experiment compared to information in your trees and events. Calculate the percentage yield given the following information. You can see the information there. From the result obtained, we were able to isolate purine alkaloids from tea using various separation techniques, and also confirm the presence of purine alkaloid in the isolate using Murray test. These are the references: your trees and events pharmacognosy textbook and your workbook 3 manual from the Department of Pharmacognosy. Thank you for your attention.